About 15 seconds until we go live, and Nikki, you are unmuted. Going live in 5, 4, 3, 2.
Hello, everyone, and welcome to today's The Scientist webinar. I'm Nikki Spahich, Scientific Technical Editor for The Scientist, and I'll be moderating our discussion. Today, Dr. Linda Saif will give a historical overview of SARS spillovers from animals to humans, and Dr. Neville Sanjana will describe recent work on a SARS-CoV-2 spike protein variant that increases human infectivity. We'd like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the speakers will address these during the Q&A session following the presentations. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. We'll try to address as many of these questions as we can during our Q&A session. The webinar will be archived on the Scientist website. We'll send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsors. Cynobiological is an international reagent supplier and service provider. The company specializes in recombinant antigen production and antibody development. They are the first company in the world to produce SARS-CoV-2 viral antigens. To support the development of immunodiagnostic assays, Cynobiological has developed a comprehensive collection of antigens and antibodies for SARS-CoV-2. Acro Biosystems is a leading manufacturer of recombinant proteins and other critical reagents to support the development of target therapeutics and diagnostic kits. To facilitate research on SARS-CoV-2 and help fight against the COVID-19 pandemic, Acro Biosystems has developed more than 60 SARS-CoV-2 related products, within which about 2/3 are recombinant SARS-CoV-2 antigens with multiple tags and species mostly expressed by HEC293 cells, covering all the critical targets of SARS-CoV-2. BPS Bioscience is a US-based original manufacturer with an innovative portfolio of over 4,000 products to assist in your drug discovery projects. As a primary manufacturer, BPS can supply products in large-scale bulk quantities and welcomes inquiries for contract and custom manufacturing within a variety of research areas. 10x Genomics builds solutions to interrogate biological systems at a resolution and scale that matches the complexity of biology. Their rapidly expanding suite of products, which includes instruments, consumables, and software, have enabled customers to make fundamental discoveries across multiple research areas, including cancer, immunology, and neuroscience. Our sponsors have provided us with some helpful resources related to today's webinar. We've posted these in our handout section, located on the right side of your screen. You can access and download these documents at any time during the webinar. And with that, let me introduce our first speaker. Dr. Linda J. Saif is a distinguished university professor at The Ohio State University in the Food Animal Health Research Program and the College of Food, Agricultural, and Environmental Sciences at the OARDC and the College of Veterinary Medicine. At OSU, she is the co-director of the Virus and Emerging Pathogens Program in the Infectious Disease Institute. She is an elected member of the U.S. National Academy of Sciences and the National Academy of Inventors. Dr. Saif is a virologist and immunologist whose coronavirus research spans 4 decades. Her lab focuses on diagnosis, pathogenesis, immunity, and vaccines for animal coronaviruses and their interspecies transmission. Dr. Saif worked with the WHO and CDC during the Severe Acute Respiratory Syndrome, or SARS, outbreak, and with the Ministry of Agriculture in Saudi Arabia on the Middle East Respiratory Syndrome, or MERS, coronavirus in camels. In 2015, she was the first woman to receive the Wolf Prize in Agriculture. In the Novel Experts Coronavirus Global Expert Landscape, she is listed in the top 10 in coronavirus vaccine publications. Let's make sure your slides are up and running. Okay, Dr. Saif, take it away.
I'd like to thank the scientists for supporting these webinars on COVID-19. And also, Megan, thank you for your technical assistance. So today I'm going to talk about emerging coronavirus of humans and animals, their origin and interspecies transmission. So many of you are familiar now with coronavirus since COVID-19. It's been on the news a lot of the time. The coronaviridity, there are 4 family with 4 genera, alpha, beta, gamma, delta. They're positive synths, enveloped, single-stranded RNA viruses, one of the largest RNA viral genomes. And the surface spike encodes the virulence factors and also the neutralizing epitope. So it's the spike that attaches to the host cell receptor. And if we make antibodies that are neutralizing, they will block this interaction. It infects mammals and birds, and it mainly causes respiratory and enteric disease. So the COVID-19 emerged in China about 7 years after the Middle East respiratory syndrome emerged in the Middle East. It occurred in December or maybe late November 2019 in Wuhan, China. Very shortly thereafter, the whole genome was sequenced. It was found to be a new beta coronavirus B related to SARS, 80% nucleotide identity, but closest to a strain from bats, 96% overall nucleotide identity. By the end of January, WHO had announced the virus was a public health emergency. And as of yesterday, the pandemic, there have been over 20 million cases, about 750,000 deaths in 216 countries. So it spread to the US in January, February 2020. And now we have over 5 million cases and over 166,000 deaths in 50 states. So this slide summarizes something about bats and birds, which are the natural ancestor reservoir for the coronaviruses. So we have bats as the ancestor coronavirus reservoir for most of the alpha coronaviruses. And we have a number of species here. We also have host range mutants, meaning that these viruses co-circulate in dogs, cats, and swine. I'll talk about that a little more later. And then we have 3 different clades of beta coronavirus. The A clade that has a large number of domestic livestock, wildlife, rodents, and humans. And we've had spillover from this into humans. And then we also have the Delta coronavirus from avian reservoirs that infect mainly avian species, but also now some mammalian species, including pigs. And the Gamma coronavirus that mainly infect avian species. Sorry. So what we see is these have spilled over for the alpha coronavirus, the common cold 229E, from bats to camelids to humans. We have the spillover from bovine coronavirus to humans of the OC43 common cold coronavirus. And then more recently, we have the clade B, the SARS coronavirus from bats through intermediate civet cats to humans, SARS coronavirus 2 that we're going to talk more about from bats, possibly to an intermediate host or directly to humans. And finally, the MERS coronavirus, a spillover from bats through an intermediate host, camelids, to humans. So there are 7 types of coronavirus associated with human infections. We know that the common cold coronavirus are endemic and the population has variable degrees of immunity. These include the alpha coronavirus 229E NL63 and the beta coronavirus OC43 HKU1. And now, more recently, we're having the pandemics associated with pneumonia mainly in a completely naive population with no immunity. And this was first with SARS, and then MERS, and then more recently, SARS coronavirus 2. So we see that the SARS and SARS coronavirus 2 are in the beta coronavirus clade here, separate for SARS-2 from SARS. And then we have the MERS in the clade C. We have bat coronavirus in clade D, and then A is the 2 common cold coronavirus. Now, they all have a similar genome structure. There are usually 4 main structural proteins, the S, the spike, the envelope, the membrane, and the nucleocapsid. Some, like HK2U1, have a hemagglutinin, as does bovine coronavirus. And then the others have similar organization, except these numbers here in purple. These are different accessory protein open reading frames, and these vary depending on the virus strain. We can see a lot present in SARS and coronavirus 2 and SARS coronavirus. These are thought to be important in terms of blocking innate immune system, so the virus can replicate in the host cells. And I'll show you some evidence that they appear to be a factor in terms of alterations that lead to interspecies transmissions. So the questions I'd like to address today are, why are the coronavirus so diverse, and how do they emerge and evolve to infect new hosts? Which coronavirus crosses species barrier? What are the animal reservoirs for SARS and COVID-19? How are they transmitted from animals to humans? What are the unknowns or gaps for SARS coronavirus 2? Just mention a few. And then some of you may not be aware of this about coronavirus causing major global outbreaks in animals, similar to the SARS pandemic in humans. So I'll discuss that and something about the origin of these epidemic strains of animal coronavirus. So why are coronavirus so diverse, and how do they emerge and evolve to infect new hosts? So I already mentioned they have one of the largest RNA genomes. Even though there is an endonuclease that has a proofreading mechanism for coronavirus, they still have a high error rate, especially compared to DNA viruses, so mutations are common. The coronavirus occurs as a quasi-species or swarm of virus in a population, and this may explain what I'll talk about in a subsequent slide, the origin of interspecies variants that have altered tissue tropism. And the example I'll talk about is the swine coronavirus, enteric transmissible gastroenteritis, and the naturally occurring respiratory variant, porcine respiratory coronavirus. We also have examples with feline enteric strains that then mutate to cause a systemic disease in cats called feline infectious peritonitis virus, because they switch the tropism from enteric to macrophages. Also, a major factor is that these coronavirus have high recombination frequencies, and this can lead to the emergence of new strains or new zoonotic strains. So I'll first start with some examples of interspecies transmission of alpha coronavirus. So I mentioned earlier, swine, cats, and dogs have alpha coronavirus, and these are thought to be host range mutants of an ancestral coronavirus. They share the same receptor, aminopeptidase N, and this receptor is also shared by the common cold alpha coronavirus 229E. The interspecies recombinants occur frequently among cats, dogs, and swine. And I'll give an example here. The canine coronavirus type 2A is the ancestor of the TG in swine. And this apparently happened by mutations in the spike, deletions in the open reading frame 3B, and then a non-functional complete loss of open reading frame 7. We also see that even more recently, the canine coronavirus 2, there have been co-infections of this in terms of canine and dogs, the canine coronavirus 2, and a TG-like strain, presumably from pigs. And this led to a new variant in dogs, the canine coronavirus 2B, that has a different pathogenesis. And this was a recombinant with just a section of the spike here acquired from the swine coronavirus. So I give an example here of interspecies variants with coronavirus tissue tropism changes. A good example is the TGE that went from enteric tropism, infecting only the intestinal tract, to a respiratory tropism, the porcine respiratory coronavirus. So the TG was first described in the 1940s, and this virus was very stable until independently in Europe and the US, we had the emergence of a porcine respiratory coronavirus variant of TG. So this virus was identical to TG except for several changes here. This lost 621 to 682 deletion in the N-terminus of the spike. And interestingly, in the strains, this deletion varies in size in all the different PRCV strains. There was also loss of functional open reading frame 3, so there were deletions or insertions here. So TG and PRCV, they still share the same receptor binding domain, and they bind to the APN receptor. But it's thought that tissue tropism changed due to the loss of sialic acid binding to gut mucins by the PRCV spark. So this is a sialic acid binding region. And interesting, this loss of enteric tropism and virulence, PRCV is now widespread in swine, and it's acted as a naturally occurring TG vaccine. So we don't see TG that often in swine in terms of a virulent infection. PRC respiratory infections are also interesting because they mimic mild SARS coronavirus 2 infections. The PRCV infects the epithelial cells of the upper and lower respiratory tract and type 1 and 2 pneumocytes. Here are the cells lining a bronchiole. Most infections are mild or subclinical. It's referred to sometimes as walking pneumonia, and some reports as high as 50% of asymptomatic COVID-19 cases may show evidence by CT scan of damage to the lungs. And it also causes an atypical pneumonia in a majority of pigs that resembles the SARS coronavirus 2 lesions. So I'll give you now an example of interspecies transmission of beta coronaviruses. So these were early studies that we did in 1995 and later in 2006. And these were some of the first studies to show that coronavirus cross species barriers and that coronavirus from wild animals can infect domestic animals. So we used the notobotic calf as a model here because it doesn't have any other pathogens or microbiota. We orally inoculated the calves with these enteric coronavirus that we isolated from captive wild ruminants, sandbar deer, white-tailed deer, or waterbuck. And we also had a strain from a child that was over 99% identical to the bovine coronavirus strain from Dr. Zhang. So after we inoculated these calves, they all developed diarrhea, they all shed virus fecally, and they all seroconverted with neutralizing antibodies to bovine coronavirus. So we concluded that coronavirus from wild ruminants or humans can experimentally infect young calves. And so this was evidence of the interspecies transmission among these species. We also did the sequence analysis, the full genome sequence analysis. These are the wild ruminant strains here in these black areas. So we can see that overall, there is 98.4 to 99.6 nucleotide adenogeny in the wild ruminant strains compared to especially the more recent bovine coronavirus strains. So this slide depicts that spillover and emergence of new coronavirus has occurred repeatedly in both humans and livestock. So we're very familiar now with SARS coronavirus spillover from bats to civet cats to humans in 2002 to 2003. Almost a decade later, we had MERS arise in the Middle East from camels to from bats to the intermediate host camels. And then most recently, the spillover of SARS from ancestor bat hosts. We don't know intermediate hosts or directly from bats to humans. So one thing that's interesting about this SARS coronavirus was not the first coronavirus transmitted from animals to humans. And what we have are these SARS coronaviruses that are associated with the common cold that also spilled over historically, some from a bat reservoir to humans almost a century ago, and then from bats to camelids to humans in the 17 to 1800s. And then more recently, 1898, this is all by molecular clock analysis of the genomes, likely from cattle to humans for the OC43. And I'll talk later about some of these spillovers that have happened recently in animals in swine. So another interesting point that the majority of these spillovers, the intermediate hosts, were ruminant species and camelids. So now we know that there are a number of coronavirus with broad host ranges. So SARS, coronavirus 2, SARS and clade B, MERS and clade C, bovine and clade A are promiscuous. So all of these coronaviruses can infect a number of species, both naturally and experimentally. And SARS coronavirus then we know infects humans. There are related strains in horseshoe bats and pangolins. We know there's been some evidence of infection naturally or experimentally in cats and felids, the tigers in the Bronx Zoo, in mink in the Netherlands, Denmark, and Spain, and occasionally in dogs. Experimentally, it can infect ferrets, monkeys, hamsters, and mice. So what are the factors that influence interspecies transmission of these coronavirus? One is exposure. When we have close contact with infected animal with infectious secretions or fomites, and this mainly will occur at the wildlife, domestic animal, or human interface. And many of these are associated with bat reservoirs or the ancestral bat host for coronavirus. This can happen in live animal markets, as we saw with the civet cat and SARS spillover, where there are multiple species, unsanitary conditions. And for swine coronavirus, porcine epidemic diarrhea virus, it's thought that this virus entered the U.S. possibly from fecally contaminated feed sources from China, and we can have also environmental contamination. Another factor is the stability or dose of the agent in the infectious material and the transmission route. There may be a greater efficiency of combined aerosol and fecal oral transmission. Bovine coronavirus, potentially SARS and MERS, are pneumoenteric viruses infecting both the upper and lower respiratory tract and intestine, and potentially for SARS coronavirus 2. Viral attachment and entry with receptors and co-receptors are critical for the first ability of the virus to enter a new host cell. And interestingly, bovine coronavirus has an influenza C-like hemagglutinin, shown here, this inner shorter layer of projections on the surface that historically was acquired many years ago in a recombination event with an influenza C-like virus. And then, like SARS coronavirus, the SARS coronavirus 2 binds to the ACE2 receptor. It's reported to have a higher binding affinity than for SARS, allowing it presumably to transmit more readily among humans. And then it's also been shown to have a new polybasic furin cleavage site that may play a role also in transmission or pathogenicity. So we know that SARS coronavirus 2 is a zoonosis. And the first observation were that animals in live animal markets in China had antibodies to SARS coronavirus 2, but no disease. And as far as the animal host, it was found mainly in these mass palm civets, but also raccoon dogs. The virus from these mass palm civets was over 99% identical, nucleotide identical to the human SARS in the markets, but wasn't found in the wild and farm civets. So these spillovers were occurring in the marketplaces. And after the SARS outbreak, the sequence data confirmed that certain bats harbor SARS-related coronaviruses. And then more disturbingly, it was found again after SARS that some bat strains actually can use the human SARS coronavirus 2 receptor ACE2. And so this certainly worried people working on coronavirus and interspecies transmission, whether bats could play a direct role in transmission to humans. And this led to the review paper where Dr. Ching and Yuen indicated that SARS-like coronavirus in bats could be a time bomb. So we know what happened next. We had the emergence of COVID-19. And we don't know if this is for certain a zoonosis. The observation is some early cases had contact with animal markets in Wuhan, China, about 2/3. We do see from the sequence data that bats are the likely ancestral host. There was the prior transmission reported and based on serology, a bat SARS-like coronavirus to humans in southern China with about 2.9% seropositive. And then 2 bat coronavirus at least have been reported that are related to SARS coronavirus 2. We have the RATG13 strain sharing 96% overall genome similarity. And then we have this RMYN02 strain that shares 97% nucleotide identity in open reading frame 1A, 1B, the replicase gene. And interesting, here is an example of a naturally occurring furin cleavage site present in a strain from bats. So is there an intermediate host besides the bats that transmitted the virus to human? Some suspect the pangolin or other infected animals in wildlife markets, but the pangolin overall has 91% nucleotide identity with SARS coronavirus 2 compared to this strain of bats. and compared with the civet cats that had greater than 99% as an intermediate host. Another concern is the occurrence of the virus and spread of SARS coronavirus 2 in mink farms. There are many, actually China leads the world in the production of furs, so there are many mink farms in China, or whether there might be spillover from a related muscalid species. So another concern now are whether pets and mink could act as secondary hosts. So there is evidence that cats and dogs were infected in households of COVID-19 cases. This was reported from a group in Hong Kong in Nature. And it's also been shown in 3 different studies that cats could be experimentally infected with SARS coronavirus 2. The cats shed virus. They showed variable respiratory pathology. And they seroconverted, and they were able to transmit virus directly or indirectly to contact cats. However, the dogs were only marginally susceptible, but they seroconverted. So in the heavily dense COVID-19 human case regions of Italy, a survey was done recently, and they found that 3.9% of household cats and 3.4% of dogs seroconverted, but this percent was much higher if these animals were present in the household of a positive case. And then I mentioned about the natural infections among mink, and a survey was done in seropositive stray cats found in the vicinity of infected mink farms, and about 30% were seropositive in the cats in the vicinity of the mink farms in the Netherlands. So now the situation with mink. There have been major outbreaks in farm mink in the Netherlands, also some reports in Denmark and Spain. It's been shown there's very rapid indirect transmission among mink. They're in cages and they're not in direct contact with mink in the next cage. The mink showed mild to severe respiratory signs. They shed virus, mainly in nasopharyngeal swabs, but also in some in feces. They developed interstitial pneumonia of those that were post-mortem and had died, and most seroconverted. So the problem here was for the government in the Netherlands, there were 2 suspect cases of transmission from mink to animal caretakers. Now, the first cases among mink were thought to have originated from the animal caretaker transmitting the virus to the mink. But later cases, there was suspect transmission based on sequence data and the sequences of virus circulating in the area from the mink to human caretakers. So because of that, almost 1 million mink have been called in the Netherlands and Spain. So the concern here is, will pets or wildlife such as felids or mustelids become a reservoir host for COVID-19? So I'll just summarize a few of the unresolved questions for COVID-19. What was the origin? How was it transmitted to humans? And under what circumstances does interspecies transmission occur? Is there an intermediate host that transmitted the virus to humans? Will humans spill over into other animals, create a new host reservoir for COVID-19? What is the role of asymptomatic or pre-symptomatic infections? Why is there enhanced disease severity in the elderly or those with comorbidity compared to children? How does the virus pathogenesis and the host response or the immunopathology influence disease progression, severity, and outcome? And then we still don't completely understand how the viruses are transmitted. The role of aerosols, non-respiratory routes, saliva, feces, if there's any chance of foodborne routes or incidental hosts, such as pets, that may play a role in SARS-2 transmission. So just the last couple of slides, I want to summarize some data. As I said, we also have major epidemic outbreaks of these coronavirus diseases in swine. So the first example is porcine epidemic diarrhea. The virus occurred in Europe in 1977. It spread in Europe and then essentially it disappeared, mainly disappeared other than a few cases in a few countries. But then the virus moved to Asia and China. And in 2013, the virus appeared in the US for the first time, and the origin was thought to be China 2012 strains that were highly similar to the strains in the US. It caused a fatal diarrhea disease in baby pigs. Almost 100% of the baby pigs on the farms died. So this occurred just like the COVID-19 in humans in a naive pig population. 8 million pigs died in the US, and now the virus is endemic. Another recent example is the porcine Delta coronavirus, and this is in the Delta coronavirus clade. It was first described in swine, Asian leopard cats, in samples surveyed in Hong Kong. In 2014, it first appeared in the US. It caused diarrhea in baby pigs like PEDV, but it was less fatal. And the origin is mainly in avian species that the virus circulates. The porcine Delta coronavirus is related to a sparrow coronavirus, but also Asian leopard cat, a mammalian coronavirus. These Delta coronavirus use APN receptor like TG, like 229E, and we showed in vitro it can infect swine, chicken, and human cells in a collaborative study with a group in the Netherlands. And also it cross-infects calves experimentally and turkeys experimentally in studies in our labs. So this just shows the potential transmission here from avian species to mammalian species and across other species barriers, including back to avian species. So this raises the question, will Delta coronavirus be the next emerging coronavirus in humans, because it has some of the characteristics that we've seen for the emergent bat coronavirus in humans. Another more recent example comes again from China, and this is the swine acute diarrhea syndrome, or SARS, emerged in 2017 in China. It caused diarrhea in baby pigs. It killed almost 25,000 swine in southern China in Guangdong province. The origin, it's most like alpha coronavirus, a high nucleotide identity detected in rhinolopus bats in Guangdong, and these are the same bat hosts for SARS. And up here you can see these are the red ones, are the farms with the SARS infections, very similar to the bats that were captured and sequenced their coronavirus sequence from the same area. And the spike is thought to be a recombinant between alpha coronavirus and an unknown beta coronavirus, which is another interesting aspect. So then to summarize, and our conclusions is that coronavirus continue to evolve and emerge globally as a cause of fatal respiratory and enteric infections of young animals and humans. They're genetically diverse with frequent mutation recombination. Some beta coronaviruses, SARS coronavirus 2, MERS, bovine coronavirus, have broad host ranges, and some of these, such as bovine coronavirus, can be zoonotic. The epidemiology and zoonotic transmission of SARS coronavirus 2 infections are unclear. And unfortunately, there are no pre-existing vaccines that have been developed for the respiratory coronavirus infections in mammals. So this includes the respiratory bovine coronavirus and the porcine respiratory coronavirus. But there is a vaccine for infectious bronchitis, mainly an upper respiratory tract infection, and infection of the bronchioles in avian species. So we're left with the concern, WHO's concern for the next pandemic labels this disease X, and it appears not to be a case of if, but when, based on all the factors I showed you for future coronavirus transmissions to humans. So finally, in terms of coronavirus interspecies transmission, just to summarize, we have bird reservoirs, the Delta Gamma, with recent spillover from presumably an avian species into swine in the US. We have bat reservoirs, the alpha, beta, B, and C clade coronaviruses, the ruminants and potentially rodents for beta A. And what these species have in common is that there are multiple species. There are thousands of species of bats and also avian species that they congregate in high density populations. There's interspecies transmission among different bat species in the caves, and they're migratory. So they can spread the coronavirus outside of a certain very regional area. Once coronavirus then adapt to new hosts with interspecies transmission, they may establish and expand within the new species so that then we set up the situation for interspecies transmission, as I showed you, for coronavirus circulating dogs, cats, and swine. And this may establish a new reservoir community. So the question is whether this will occur for COVID-19. So finally, I'd like to acknowledge my lab, Dr. Silva lab, all the people who have worked on various aspects of these coronavirus infection, Dr. Chiang Wong lab, Dr. Scott Kinney lab, that's been working on the porcine Delta coronavirus, and then assistance with infectious clones from Dr. Ralph Barrick's lab and our grant funding. And I'll be glad in the last part of the our symposium to answer any questions. Thank you.
Thank you for that presentation, Dr. Saif. As a reminder to our audience, you may ask questions at any time using the Q&A tab. Our panel will have a chance to address these after our next speaker. That next speaker is Dr. Neville Sanjana. Neville Sanjana is a core faculty member at the New York Genome Center and assistant professor in the departments of biology and neuroscience and physiology at New York University. As a bioengineer, Dr. Sanjana creates new tools to understand the impact of genetic changes on the nervous system and cancer evolution. His lab has harnessed high-throughput gene editing to pinpoint which regions of the genome are involved in diverse diseases. Dr. Sanjana is a recipient of the Presidential Early Career Award for Engineers and Scientists, the AAAS Watchell Prize for Cancer Research, the NIH's New Innovator Award, the Cancer Research Institute Technology Impact Award, the DARPA Young Faculty Award, the Kimmel Scholar Award, the MRA Young Investigator Award, and also is the LexGen Family Investigator of the Brain and Behavior Foundation. Dr. Sanjana holds a PhD in Brain and Cognitive Sciences from MIT, a BS in Symbolic Systems, and a BA in English from Stanford University. Let's just make sure the slides are up and running. Okay, Dr. Sanjana, take it away.
Great. Thank you so much for having me here. I want to thank Nikki and the scientists for this really nice invitation. And I first want to say that it's really an honor to speak after a pioneering virologist like Linda Saif. So my wife received her PhD in Dr. Saif's lab, and she always describes it as one of the most wonderful scientific environments. So it's really just an honor to be here with Dr. Saif today. So great. I'm going to tell you about some recent work that we've been doing in our lab to really examine just one tiny part, I would say, of the SARS-CoV-2 virus, which is this spike protein that it uses for entry. And I think the previous presentation really, I think, sets the stage very nicely for us to now delve down into some of these details of viral entry. So the work I'm going to tell you about today was a team effort, but it was largely led by one very talented postdoc in my lab, Jarko Danilowski. And if you're interested in actually seeing the preprint, you can go to this to bioRxiv with this link that's right here. So one way that we and I think a lot of other folks started to get interested in this mutation in the spike protein, and again, the spike is really the protein that gives coronavirus its name. It's what makes the corona or crown of the virus. It's this spiky little thing that you've seen in all the illustrations of coronavirus. And as we just learned, this virus emerged in late 2019. And because of the somewhat error-prone nature of the viral polymerase, that virus has not stayed constant over time. It has accumulated some mutations, it changes. And one of those mutations that has attracted quite a bit of attention emerged probably in maybe late January, early February 2020, which changed just one bit of the protein, one amino acid of the protein spike. And what I'm showing you here is kind of the original version of the SARS-CoV-2 spike, which is the one that was first sequenced from Wuhan, China. That has this D614, so those are the green dots here. And then you can see in this kind of this diagram that shows a few thousand different patient isolates, you can see that all of a sudden in early February, perhaps, you see the emergence of patients who carry a different variant, G614. a change in the spike protein. And what you can probably observe here is that that goes from being at a very, very low frequency in the population to really growing and taking over. And here's just a nice graphic that I've taken from Science magazine, and it just shows how quickly this takeover has happened. That we've gone from pretty much 100% of this D version, D614 version, in just the span of a few short months to nearly 100% of the G614 version. And of course, that leads to a lot of questions. What is this variant doing? Is it being selected for in some way? When something sweeps a population like this, we think there must be some natural selection at play. It seems less likely that this is, just some random genetic change. And before getting into actually the evidence, looking at kind of what we did in the lab, looking for evidence of increased infection, we were really, as we started to get into this, we were really impressed to see others like Tim Cardozo, who's my colleague here at NYU, who did this really nice study where he looked across different countries in the February-March timeframe when you had both the original variant and the new G614 variant circulating. And he noticed this really nice trend. And here I've replotted it with a larger data set that includes April and May. But the trend was basically that the higher the prevalence of this G form of the virus, the higher the case fatality rate in that country. So every dot here on this plot is a different country plotting the prevalence of the mutation and the fatality rate from COVID-19. And you can see that there is some relationship with the prevalence. I think kind of even more direct evidence in patient populations comes from two different studies, one done in the UK at the University of Sheffield, and another one done at the University of Washington here in the United States, in Seattle. And here what they did is they had a few hundred COVID-19 patients who either had the original variant, the D variant, or they had the mutation, the G variant. And they did this nasal swab test that we're all very familiar with now, this thing that kind of is this long Q-tip that samples the upper nasal cavities and then is read out by something called qPCR or quantitative PCR. And it basically just measures how many viral copies they are. And what was really interesting is that these two sites with very different testing protocols, different ways of different reagents, things like this, they had this very, very consistent difference that they noticed. So you can see here that the threshold cycle for the qPCR is always lower in the case where there's this G variant. And that's true whether you look at the data from the UK or you look at the data from the US. And what that means for those who might not be super familiar with qPCR data is that there is an increased prevalence in folks who have the G variant. Because the virus is being detected earlier in the qPCR, there's increased prevalence of the virus. There's an increased viral load. And if you look at it across these two different sites, and you can see that there's a systematic bias in the qPCR protocol, meaning that the cycle numbers are lower in the University of Washington versus Sheffield. That's just a technical difference. But if you look at the delta, the difference between the D and the G variant, it's very, very similar. That's the third panel here, very similar, whether it's the University of Sheffield or University of Washington. And so it's really nice to see that across these two different sites, you see this very consistent difference. And what this consistent difference implies is that there's threefold more virus circulating within folks who have the G variant, meaning that it's not only perhaps more infectious between different from one person to another, but even within a single person. If you think about it, the virus it doesn't just infect you, it goes from cell to cell to cell within a single body. And that's even within a single person, there's greater infectiousness, greater viral load. So with all this kind of in the background, we decided we wanted to see if we could find direct evidence to really measure this across different human tissue types. We know that COVID, even though there's a lot of symptoms in the lung, that it actually infects many other cells in the body and has a very diverse set of symptoms. So we took human cells from lung, from liver, and from colon, And what we did is we took a virus that we commonly use here in the lab that we understand very well, and we coated these viruses either with the SARS-CoV-2 D variant, the original variant, D614, or we coated these viruses with the G variant, the one carrying this mutation. And we looked to see, is there a difference in how well these infect these different human cell types? And one thing we did is we engineered these viruses so that they would carry, as a payload, they would carry a fluorescent protein. So they basically turn the cells green, makes it very easy to see these cells. And the question is, do you see more green cells in this case of the mutation, the G614 spike, than you would see with the other spike or without any pseudotyping at all, any coat protein? And indeed, that's exactly what we saw. So here on the left panel, I'm showing you data just really from one cell type, one lung cell type. And you can see that if you look here at the amount of GFP, at the amount of green fluorescent protein, you can see when you add the spike pseudotype, it increases. That's the green right here, the green bump. And if you add the spike protein, but just make this one small mutation to G614, you can see a huge shift, a log order of magnitude increase in the amount of GFP, much more transduction of these cells, infection of these cells. And this is true. I'm showing you, kind of the summary data now for not just lung, but also these HUH 7.5 cells, which is a liver cell line, or these cacao-2 cells, which is a human colon cell line. So really across a broad variety of tissues, when we do this very carefully controlled experiment where all we're modulating is this one mutation on the spike protein, you can see this big change, about 2 to 8-fold increase in infection of these cells. And one very important control that I want to just show to you to prove to you that it's not due to something else is that there's not really a difference in viral titer. We, of course, make these viruses kind of in parallel with either the D614 or G614. And one thing we can do to make sure there's no difference in the number of viral particles produced is we can do, again, a qPCR-based assay to read out the number of genome copies. And we have this here with a couple different primer sets. What you can see is that the ratio of wild type to mutant in our different viral preps is 1. It's the same number of virions are being produced, but they're so we can be confident that the difference in infectivity is really due to the difference in the spike protein. Okay, so that's interesting to show that this mutation is making these virus particles more infectious. The natural question is how? How is this happening? And for that, we're going to delve a little bit into the structure of the spike protein. So, of course, the spike protein has a 3-dimensional structure that I'm going to show you in a few slides, but here's it kind of displayed in a linear format from end to C term, from beginning to end. And one thing that you can notice if you look to see where this mutation is in the protein is that it lies somewhat close, though not inside of, the receptor binding domain. And the receptor that SARS-CoV-2 spike uses with human cells is now well established by many groups to be the ACE2 receptor. And I think, let me see if the next slide has this. Yeah, so here is something that this is some data from earlier this year showing that really only cells that express ACE2, so at the top row is cells expressing ACE2, the bottom row is cells that don't express ACE2. Only when you have cells that express ACE2 can you have infection by SARS-CoV-2, and that's detected here by using immunofluorescence against the N protein, the nucleocapsid protein of SARS-CoV-2. So what we were interested in is, well, if this is close to this receptor binding domain, maybe the reason that this mutation in the spike protein creates a more infectious virus is that it just binds its receptor on human cells better, right? It needs to bind the receptor to get into the cells. Maybe that's what's happening. And so here we collaborated with Jira Baba's lab at NYU Med School with a talented graduate student there, Juliana Ilmain. And with Jira and Juliana, we purified either the mutant or the wild type spike protein and we immobilized the ACE2 receptor on a very high resolution interferometer. And we looked to see what are the binding kinetics of these two different spike proteins as we run them over the ACE2 receptor. Can we see a tighter binding interaction with the mutant spike than we would with the original wild type spike that came from Wuhan? And here, when we do this high resolution assay and we fit some dissociation constant here, we really see no significant difference. They're within the kind of error range of measurement for this kind of measurement. Both have low nanomolar range binding affinities, whether it's the wild type or the D614G mutation. So it really doesn't seem likely that the difference is due to increased binding of ACE2. Okay, so if it's not that, how we were still kind of puzzled, how does this actually work? And so there we started to examine another property of the spike protein, which is that this protein is processed in a few steps as it moves the virus from initial binding of the ACE2 receptor on human cells to actually entry into those cells. And so you can see here that I've labeled these fragments, these proteolytic cleavage sites, as S1 and S2, and then there's a further cleavage of the S2 into this S2 prime fragment. And that leaves this thing called the fusion polypeptide exposed, and that's what really sticks into the target cell membrane. It's a hydrophobic region that can get into that lipid bilayer of the target cells. And so, just to give you more of a 3D structure, because these proteins exist as these beautiful sculptures here, and so whether you look at kind of SARS-1, the original SARS from about 20 years ago, or the SARS-CoV-2 spike, they have a very similar structure. And so you can see the difference between the S1, which is colored here and on the top, and the S2 subunit, which is in gray on the bottom. And so the S1 subunit is what's responsible for that initial interaction. It sticks out from the virus. It's the one that kind of touches the ACE receptor originally, and then it pops off. And the S2 is kind of like a needle that can then insert into the host cell membrane, into the target cell membrane. And so here's kind of what that looks like in a little bit of a different view. This is a very beautiful paper that was recently published by another group in Science. And you can see that, that on any particular SARS-CoV-2 virus particle, you have a mix of spikes that are both in this kind of full-length form where they have the S1 and S2 complex intact, that's here in purple, and spikes that have lost their S1 fragment, and they're just this long S2 needle. And so there's some mix of both of these on any particular viral fragment. So one question that Jarko and I had is, well, maybe it's the distribution, the difference between how many are full S1, S2 complexes and how many have already lost the S1 fragment that might be responsible for this difference in the mutant form of the virus versus the original form of spike. And just to kind of show this in action, I think this diagram is really nice. So here you can see on the left side, you can see the full S1, S2 spike here. It interacts on the top with this ACE2 receptor. Then after binding with the ACE2, which helps it bring the membranes close to each other, the membrane of the virus with the membrane of the cell close to each other, it kind of goes away. And then what you're left with is just this S2 fragment that then enables the fusion and this post-fusion state and allows the viral genome, the viral RNA, to get into the target cell. So we wondered whether it was something about, are you missing one or the other of these forms? Right. And so it's one more thing to show you here before I jump to the data is it's important to know that that ACE2 is very important for viral infection, so that if we block ACE2, specifically if we block ACE2 for SARS or for SARS-2, it really isn't able to jump into the cell. It critically requires having ACE2 present. And that's true looking at this data from Stefan Pohlmann's group, and also looking at data from our group, where we really see a huge difference with ACE2 and being able to enter into the target cell. And so here, what did we actually find? So what we did is we overexpressed these two forms of spike. We have this kind of cloned into a uniform vector, a very standard expression system. And we overexpress these in human cells. And we have an infinity tag on the C terminus of this protein. So we can pull it down and look to see how much of this is in the full length state, which is shown up here as S on the blot on the left, and how much is in this process state, either as S2 or S2 prime. And what you can hopefully see here is that with the mutation, the G form of the spike, we see a decrease in premature cleavage down to that S2, that fully needled state. And we repeated this several times. We were very amazed that this one amino acid mutation could just make such a big difference. And indeed it does. So if you look here on the right, you can see that there's really this huge increase in cleavage in the D form. So it's easier to cleave that form into these fragments, whereas the mutant form kind of stays intact a little bit better. It's able to stay as a full protein. And of course, these spikes are produced in a host cell, and then they have to go emerge from that host cell and infect a target cell. So you can imagine that if there's some premature cleavage events that happen in the host cell, by the time they get to that target cell, they might just not be that effective and able to do their job if they're missing that ACE2 binding domain. I think there's actually, I should say, a lot more biochemistry that can be worked out with this, but at least it seems likely that this is much more involved with the mechanism of how this mutation has an impact than a change in ACE2 binding. So, one thing that I haven't really gone into much detail up to now, but I mentioned it in passing, is that we've done all this kind of in a very safe viral system that we understand well, that we can use here in our lab. And, it could be quite possible that in our pseudovirus system, which is actually the guts of it are really a lentivirus, that maybe that there's something different about how the lentivirus displays the spike protein on its surface. Maybe there's just a different ordering or packing of it. And really to understand whether this variant is more infectious, it'd be really nice to see that with the real deal, with the real SARS-CoV-2 full-on virus. And to do this, we thought of a few different options. One is, of course, this is causing a global pandemic. There are many natural isolates circulating in the population that we could look at. And the other one would be to make a change through some in vitro cloning or genome editing kind of the virus itself. And so when we first considered the natural isolates, we quickly ran into a problem, which is that in all natural isolates that have this mutation in the spike protein, there's unfortunately also another mutation that's found in the ORF1b gene, this P314L shown here on the bottom. And so the problem here is that we'd really prefer an isogenic system, because if we have to have a patient isolate that has both of these mutations, then it's really hard to ascribe any differences we see exclusively to the spike mutation. They might be due, in fact, to this other mutation that's just, here on ORF1b that travels along with it. So due to this idea of linkage, that these two are linked together, it's very difficult to use natural isolates to perform a very clean experiment. So here we turned to something that's actually a little bit closer to home, which is what my lab works on regularly, which is genome editing and genome engineering using CRISPR systems. So we considered doing this two different ways. One was to clone the full viral genome and just insert this change, but the virus is actually quite large. It's almost 30 kilobases, and that's difficult to do with traditional cloning techniques. The other option would be to edit this virus. And of course, many folks have heard about CRISPR technologies. It's worth mentioning that most CRISPR technologies, like the famous Cas9 enzyme, work on DNA. And this virus, the SARS-CoV-2 virus, doesn't have any DNA. Its genome is quite different from the human genome. It has an RNA genome. And we recently have developed tools to do high throughput screens using a new kind of CRISPR enzyme, Cas13, which was the focus of this article that was in Nature Biotechnology from our group 2 months ago in June. And so we did consider using a Cas13 based approach here. to target the RNA. But in the end, we actually came up with, I think, what is a simpler and very elegant approach. It's worth saying that there are several groups working on using Cas13 both as a diagnostic and as a therapeutic for COVID-19. And so I think there are some interesting possibilities in this direction. But that's not what we went with. We actually found what I think is actually quite a neat little trick that makes this a much simpler task. And of course, here to use the real virus, because we don't have a biosafety level 3 facility here at the New York Genome Center, we reached out to Ben Tenuver's lab at Mount Sinai, and specifically Tristan Jordan, who's a very talented postdoc in his lab, and it's just really been a pleasure to collaborate with them. We've had lots of great projects that have come out of this initial work. And so with Ben and Tristan, we really came up with this interesting idea, which is, as I mentioned, ACE2 is required for SARS-CoV-2 to enter into human cells. So if you have cells that don't have ACE2, it basically is not going to infect them. So what we did is we used human cells that don't express high levels of ACE2 naturally, and we co-delivered both ACE2 and a SARS-CoV-2 spike, either the D614 variant or the G614 variant. And by doing that as a co-transfection, only the cells that get the ACE2 protein are going to get this huge amount of this spike variant. And then when we put on the real SARS-CoV-2 virus, basically any viral particles that are produced are going to have whatever spike we programmed into this plasmid, either the D614 variant or the G614 variant. And so we can do this in this uniform way, side by side, and quantify the degree of cell infection. And so that's exactly what we did with Tristan. And here I'm showing you some images that he took at 12, 18, or 24 hours post-infection with this assay, which we call a transcomplementation assay, because we're delivering the spike protein in trans. And what you can hopefully appreciate visually here is that we're staining for CoV-2 infected cells using the nucleocapsid protein, the N protein stain. You can see higher levels of this, even with a very tiny amount of virus applied, MOI 0.01. And here I'm just going to show you this quantified. So again, the variant, the mutated spike, is in yellow here. And you can see whether we do a low or a high amount of virus, which is what this MOI means, a low amount or a high amount of virus. And at all different time points, 12 hours, 18 hours, 24 hours, doesn't matter what time point we look at. Every single time point, every single amount of virus we used, we always see a greater number of cells infected with SARS-CoV-2 when we're using the mutant spike versus the wild type spike. Okay, so this is this is the reality of what we're dealing with. That there's this mutation has occurred, and now it really seems to have taken over the circulating population of SARS-CoV-2. So what can we take away from this? As probably everybody is aware, there are many vaccines right now under development that have reached different stages of phase 1, phase 2, phase 3, some even towards approval. How does this impact these vaccines? Certainly, a lot of these vaccines, the development of them started even before this mutation likely existed. So there's just no way that they would even have known about it. I think the good news is that most of these vaccines are based on full-length spike protein. And I should say that most of these vaccines are based on the spike protein. So it is relevant to think about, hey, how does a mutation in spike protein affect the performance of a vaccine? The good news is that most of these vaccines, whether it's the RNA vaccine or the DNA vaccine or protein-based vaccines, most of them are based on full-length spike. So this mutation is, of course, just a single amino acid within that full-length spike protein. But when we do some analysis of this, we can see that with common MHC class 1 alleles, we can actually see that there are some reasons to be concerned that there are changes in immunogenic peptides that are previously immunogenic that are no longer immunogenic due to this mutation that's going from D614 to G614. Overall, I think we just have to test and we have to see empirically how these vaccines work. Of course, that will be straightforward since most of the population now carries this variant, but it is worthwhile to note that these can affect the generation of immunogenic peptides that our immune system can use to recognize this mutation in this spike protein. So with that, I'm just going to wrap up and give you a summary of what I presented today. So I started by showing you that in 2 different studies with 2 different groups, that the mutation, the mutated form of spike, results in a higher viral load in COVID-19 patients. And we also found that this, with the pseudovirus, causes a greater degree of infection in a broad range of human cell types, lung, liver, and colon. Our first hypothesis that, hey, maybe this works by altering the affinity to the ACE2 receptor, we tested this and we showed that this is not the case, but rather that it's more likely that it reduces premature cleavage into this S2 and S2 prime fragment and allows the virus to have more intact spikes as it approaches cells, target cells, and infects new hosts. And then to really take care of any concerns that this might be due to some artifact of the pseudovirus, we tested this with real SARS-CoV-2, where we had this very isogenic system, this carefully controlled system, where we either had the D variant or the G variant, and we showed using that indeed the G variant more readily infects human cells with real SARS-CoV-2 virus. And so with that, I'll just thank the folks who are in my group who are here on the left-hand side, and in particular Jarko, the postdoc who led the work in my group. And this wouldn't have been possible without our collaborators, Tristan and Ben from Mount Sinai, and Juliana and Gira from NYU. And also, it wouldn't have been possible without the generous support of everybody listed below who supports our lab's work. With that, thank you so much.
Thank you, Dr. Sanjana. The audience has already submitted several questions, so let's get to them. Dr. Saif, the first one's for you. We've been getting several variations of a similar question. Can you speak about the evidence behind the direct animal to human transmission of SARS-CoV-2? And what do you think about some of the claims that perhaps the virus escaped from a lab in Wuhan rather than a direct transmission?
So what I tried to show with the bat data is that there are bat coronaviruses circulating that are actually related to the SARS coronavirus 2. So one is 96% nucleotide identity. The other had regions that had similarity. The pangolin virus has high similarity in the receptor binding domain. So all things considered, based on our past experience with coronavirus, SARS, and MERS, this virus actually looks more like a recombinant between some bat and possibly pangolin and maybe other wild animal coronavirus that are circulating. So I would say that there's no evidence that this virus existed before SARS coronavirus 2. So without the virus existing that is closely related to SARS coronavirus 2, it would not be possible to escape from a lab. So I think the consensus among many coronavirologists is that likely this did evolve with a bat ancestor and then presumably was circulating in some prior form in animals and then probably transmitted to humans. I think that's the basis of the information we have now.
Great, thank you. Dr. Sanjana, what exactly is the structural basis for the D614G effect? Is that amino acid within a proteolytic cleavage site?
Yeah, that's a fantastic question. It's not within a proteolytic cleavage site, so it's not at the S1, S2 boundary or the S2 prime cleavage site. It's kind of in a disorder domain that lies between some other domains where we know the function well. But it's a great question, and I think that's something that we need probably future work to see exactly, structural studies with electron microscopy to really understand exactly how this, what is the biophysical mechanism here.
Great, thank you. Dr. Saif, what is being done currently to track coronaviruses in either animal reservoirs to control spillover? And how can we perhaps avoid future transmission to humans of other strains?
So I think this is a multi-component question. In terms of tracking, as we know, there were a number of scientists working with Chinese counterparts to track the corona-like viruses or the SARS-like coronavirus that are present in bats. It's going to be very important that continue. Unfortunately, we have not surveyed a lot of wildlife potential reservoirs, so that's research that needs to be done. And it's also important with the SARS spread to be monitoring both pet animals, as I mentioned, but also domestic livestock to be sure there's not any kind of spillover occurring from human or environmental sources. So that'll be extremely important. And so I think monitoring is one way to know what we're dealing with. And obviously we knew that there were SARS-like coronavirus in bats that could, in fact, interact with the human ACE2 receptor. But unfortunately, I'm sorry, this was largely ignored. And so in terms of research funding and follow-up. So I think we need to follow up on these studies. We need to continue monitoring bat species, wildlife, and even our domestic and companion animals to make sure that the virus is not spilling over into these species. We also have human behavior as another component. So we have the interface where humans are encroaching on the area where bats, the bat caves, there's a lot of urbanization. We have encroachment in terms of wildlife in areas where there's deforestation, there are roads that are giving humans now access to wildlife before where there wasn't that close connection. And then we have the wild animal markets. And I showed some pictures with those in where you see animals congregated together, different species, completely direct contact through the cages. And this obviously is a major, major potential for spillover from animals to humans and the consumption of these wild animals, including some consumption of bats.
Great, thank you. Dr. Sanjana, what are your thoughts on the possibility of having a universal vaccine targeting the spike protein? And also, what do you think about the vaccine that's supposedly developed in Russia?
Great. Yeah, thanks for that question. So I think, I think the possibility, the potential for a universal vaccine, I mean, I think I listen probably just to the same news that everybody else listens to. And, I saw even, I guess, just yesterday, you hear Dr. Fauci mentioning the timeline for a vaccine still to the, end of this year, early next year. I think there's a good possibility given the tremendous amount of efforts, not just on the science side, but also on the supply chain side of building up what it will take to distribute that. So I think there's a good chance. It's worth saying that the mutation rate of not just the spike protein, but the SARS-CoV-2 virus, it's high, but it's not, there are other viruses that have been much higher that we have in different ways conquered. Like a good example is HIV. So I think the chances are good for a universal vaccine. I unfortunately don't know really any of the details about the Russian vaccine, except that maybe that there hasn't been a full phase 3 trial yet. But yeah, I don't know too much about that.
So Nikki, if I could follow up on that in terms of if we're talking about a universal vaccine for SARS-like coronaviruses, as opposed to just SARS or SARS coronavirus 2, more likely we would need to target an area in the S2 where it's more conserved. And this would be at an infancy stage because there has been very little research about cross-reactive epitopes in terms of these different SARS-like coronavirus, where you could actually have a good and also a neutralizing immune response. So that would be analogous to some of these influenza vaccines, trying to make the universal influenza vaccine that are still, there's been a lot of research on that, but there's still not really a vaccine like that at this stage. So we would be at an earlier stage to be able to develop a SARS-like coronavirus, more universal SARS vaccine.
Great, thank you. Dr. Saif, what makes bats such great virus reservoirs, and are there bat coronavirus reservoirs in the United States for either SARS-CoV-2 or another coronavirus?
So there are studies, and there are some studies in the US, a lot of studies in Asia. It's mainly these rhinolopus bats that appear to harbor a lot of these SARS-like coronavirus, and that species is mainly in Asia, as I understand it. But there have now been many, many reports that different bat species harbor different alpha or beta coronaviruses apart from the SARS-like coronaviruses. So probably every bat has its own repertoire of coronaviruses, and probably many that haven't even been discovered yet. So I would say yes, probably there are coronaviruses in bats in the US, but I don't think anyone at this stage has reported any SARS-like coronavirus. As to why they're such good hosts, they have differences in their immunologic response to the virus. The virus is able to persist apparently in these species without really causing disease. So there have been essentially coevolution of some of these coronavirus historically, ancestrally in bats and bat evolution, so that they've learned to essentially live with these coronavirus. And there are many papers about the adaptations in bats that are likely what allowed them to harbor these viruses without causing disease.
Great, thank you. Dr. Sanjana, is the G614 variant observed geographically to the same degree? Is it distributed equally globally, or are there other variants observed in different countries?
Yeah, that's a great question. So initially, there was more disparity in where the mutation was observed. So initially, the kind of hotspots for it are where we think it originated, which is likely in Europe, and one of the European outbreaks that happened before it really came to the US. So it's the places that I guess where coronavirus made it a little bit later tend to carry more of the mutation. But as I said, right now, it really is worldwide. So even in China, this mutation, initially was at a very, very low fraction, but there it's grown to a large fraction of cases. I don't know exactly what it is in China, but there, every time I've looked, it's growing and growing in prevalence.
Great, thank you. Dr. Saif, given the variations in reporting standards around the world and variable performance of numerous diagnostic tests, how accurate do you think the numbers are for reported cases and deaths?
Well, I think CDC itself has said, as far as community spread, there are many more cases out there than essentially the tip of the iceberg that what have been detected. So we know that the cases, various diagnostic tests have variable sensitivity and specificity. So that needs to be kept in mind in terms of using these tests. It's possible that if one used a less sensitive test for screening, one may want to confirm it with a more sensitive test, but the dearth of tests have essentially precluded doing this. So it's apparent that for the number of cases you're detecting, especially if your test is not optimally sensitive or specific, there are probably many more cases go undetected.
Great, thank you. And now we have time for just one more question. Dr. Sanjana, based on the historical data with the virus going through the human population, do you expect that there's going to be another prevalent spike mutation coming up for SARS coronavirus 2?
I think that's a great question. Honestly, I think it's hard to predict what's going to happen in the next few months here with this. I think it's likely that we'll see other mutations, maybe not in spike per se, but even in other, the other 25 viral proteins that are there that might impact how virulent the virus is, how well it's suited to certain populations, things like this. So I do think one capability that we need to develop is a way to test these mutations in a more high throughput fashion. So what we did, we did this over the course of 2 months. I think it's worthwhile to think about investing in techniques where we can very quickly profile these mutations in different assays using pseudoviruses, using real viruses to be able to respond dynamically to any new mutations in the population.
Thank you. Unfortunately, that's all the time we have for today. If you have any further questions, please consider reaching out to the speakers directly. Their emails are shown on the screen. As a reminder, the webinar will be archived on the Scientist website, and you will receive an email notifying you when the on-demand webinar is available. I'd like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the Scientist, I'd also like to thank our speakers, Dr. Linda Saif and Dr. Neville Sanjana, as well as our sponsors, Cyanobiological, Acro Biosystems, BPS Bioscience, and 10x Genomics. Thanks, everyone, and goodbye.